Okay, so actually, now I'm working University of New South Wales from Sydney. So my talk is also focused on the irritable inflammatory bowel disease, IBD. I'm glad my previous talk already talked about what is ulcerative colitis, what is Crohn's disease, so I don't really have to repeat. And so in Australia, Australia kind of developed the country, and we have quite some instances for IBD patients. And you see in this data is from 2005, as around 70,000 Australians live with IBD. So you probably think this number is not so high, considering we have a population about 20 million. So this it's around 1% or just less than 1% patients with IBD. But the thing is, you can see the trend. Point out. Yeah, okay, so you can see the trend is going up, and now this is 2005 data, so we increase with years. And also most important things is the age of patients. They around 30, 40-ish as the peak time, so which is because they are the breadwinner of the family. If they have IBD, they probably, especially when they relapse and have to stop work, so which affect their family life. And the cost for IBD treatment is around 2.7 billion per annum. It's quite a lot cost for the community. So for the IBD treatment, as mentioned before, we also in Australia we have treatment for the reduce inflammation and also maintain remission, etc. So have 5 amino salicylic acid derivatives, and also use corticosteroids and some immuno agents, and also infliximab. For looks like 5-ASA is more effective for ulcerative colitis, and infliximab is more effective for the Crohn's disease. I'm not sure if the other country have a similar impression as well. And also, if all the treatment is not effective, have to have the surgical remove. And my research is I actually use a real human tissue. I have a human tissue bank, a colon bank, and the specimen for IBD received its surgically removed samples. Therefore, their symptoms looks like it's more severe. Okay, because I'm in the research, I like to have some models to do the IBD, and this here listed the animal model for IBD, and you can see a lot of them are some like a genetic engineered, some is chemical induced, and so for the chemical induced, you can see here for the rat and mouse, it is these 2 species most common used, and have a very common it's used DSS model or TMBS is induced in both rats and the mouse. And for the gene knockout and interleukin-10 knockout is very common. And the thing is, all these models have advantages. Okay, it's easy to do, and animal cost, of course, it's a lot cheaper compared to some other species. And also, you can see the whole body change, which is very good. However, disadvantages occur as well, especially, for example, and the experience learned, the lesson learned is this TMBS and DSS model used to develop one of the agents called the tachycanon NK1 receptor antagonist, which is really effective to treat TMBS-induced colitis. And the pharmaceutical company really invests a lot of money try to develop an antagonist to treat. And it's worked on mouse and on rats, but once used in clinical trials to treat IBD in human, it didn't work at all. Didn't work. So it's lots of pharmaceutical company has to close some of the researchers' lab and doing clinical trial for TK1. So that's the lesson learned. And also for interleukin-10 knockout mouse, it's just very hot topic at the moment for the animal models. But when we test interleukin-10 in human colon, and we couldn't detect it. So we did lots of cytokine, chemical, and beads to try to which is involving IBD. That's interleukin-10 is almost undetectable. So that's in normal control also undetectable. Therefore, we really doubt it's interleukin-10, and it's that important in human colon. All right, so, this is some words is missing, alpha and beta. Okay, this probably my Mac is not very compatible with this PC. So very recently, Scott Smith group from Adelaide University, and they actually developed a human colonic explant colitis model. What they use, actually, they use TNF-alpha and interleukin-1 beta to culture with colonic mucosa they got from cancer patients. So after, you can see after 20 hours incubation, and they actually find the damage of the mucosa, the feature is very similar, resembles to IBD you really observed in the patients. And also they found that proliferative markers as a very hugely increased. And you can see the lymphocyte infiltration occurred in this treatment as well. So that's the one model they developed. And soon after, they also use interleukin-17 to treat the tissue explant, and they found that the similar have occurred. So that's the for the human colon explant models. Now, I'm going to talk about purinergic ATP release in the intestine. Why ATP release is important and associated with IBD. All right, so ATP, the full name is adenosine triphosphate. It's very, everybody knows ATP is very important energy molecules in the body, right? So every single cell needs ATP. Without ATP, the cells will not have energy and not survive. So that's the intracellular ATP. But what we know now, ATP actually can release from cells to the environment surrounding and tissue. And first reported the ATP can be as a neurotransmitter and neuromodulator. It's by Jeffrey Bernstein. He actually thinks it's ATP. It's a neurotransmitter, can be a pure neuron, or can be co-transmitter, correlated, co-localized with acetylcholine and noradrenaline, etc. And ATP also can work as autocrine and paracrine signal molecules. So once ATP released from cells and how they work, they'll interact with the receptor. We have a purinergic P2 receptor, which can be divided into two larger classes. One is called the G protein coupled P2Y receptor. Another one is ligand-gated ion channel P2X receptor. So ATP also very easy to break down by the ectonucleotidases to ADP, AMP, and adenosine. So adenosine also work another purinergic receptor. It's P1 receptor, or used to call the adenosine receptor. So that's how they work, and then they can produce lots of numerous cell functions through P2 receptor and P1 receptor. But then we quite interested what's mechanism to mediate ATP release from cells, which channel they release from. Okay, so they can release from like a vesicle release, especially in the nerves. They have packed in the vesicle. They release from the vesicles. That's a vesicle field release. They also can release through some binding cassette or transporter. It's called the ATP binding transporter, etc. And number 3, they can release from some channels like chloride channels and KHM1 channels and connexin hemichannel and pannexin channels. So my group actually, I have a few PhD students. They actually, each of them is interested in some channels. And today I'm focused on the pannexin channel. So pannexin, probably for some of you, is quite new name. I'm not sure if you ever heard. So pannexin, its name come from, I think, their structure. When they screen from the human and mouse genome, they find one of the protein, the structure very similar to innexin, so which is the connexin or gap junction from insects, right? So from the insect species. So therefore, they think pannexin must be another form of gap junction protein and in the mammalian species. But then they find pannexin actually different from connexin because they do not form gap junction. They actually have hemichannels. They are around the cell surface and they can have a function as a ATP release channel. So that's it's being recognized as ATP release channel in many tissues. And they also recently find that they probably play quite an important role in the immune function. Okay, so when we talk about P2X, pannexin channel, I also like to talk about the purinergic P2X7 receptor. So P2X receptor have quite a few subclasses. One of them P2X7. P2X7 it's similar to the others. You can see here it's TM1, TM2 is transmembrane, but I have to have these 3 of these structures. We call the trimer to form ion channels. That's similar to the other type of P2X receptors. The difference is to activate P2X7 has to have very high concentration of ATP. Okay, not low concentration, have to have high ATP. And also their downstream signaling couples to a pro-inflammatory cascade. And they also can have generate a non-selective large pore. One of them to generate large pore is associated with pannexin. So that's the really nice study and conducted in the oocytes. So what they did is they just put P2X7 and pannexin receptor, both of them, into oocytes. So then they apply for relatively low concentration of ATP. And you can see the cells started to have something. This is, you can see the 2 sides. Here it's no transfection. This one's a transfection with both P2X7 and P2X1. So when they apply high concentration, 500 micromolar, you can see that all the yellowish color, and it starts to leak from the cells, then cause cell death. And that's the condition. You have to have both receptor and put together and apply for very high concentration ATP, and these two receptors can form really big pore. So because they're big, the contents of inside cells can leak out. So that's the so-called death channel. And a very recent studies, they already showed the Pannexin and P2X receptor, they involve in inflammation, such as they find Pannexin-1 is important for T cell activations, and Pannexin-1 also can mediate interleukin-1 beta release and caspase-1, which is the enzyme to cut the protein of interleukin-1b become active interleukin-1 beta, right? So that's happened in lung and glial cells in the central nervous system, etc. Many studies prove that's the case. And also some Pannexin, P2X7 antagonists, for example, they can inhibit IPS-induced inflammation and interleukin-1 beta release from the hippocampus. So now our question is there a role for Pannexin-1 and the P2X7 receptor in the inflammatory bowel disease? So in 2012, and Shakey's group from Canada, and they published an article in Nature Medicine, and they said it's entitled Activation of Neuronal P2X7 Receptor Pannexin-1 Mediated the Death of Enteric Neuron During Colitis. So that's the thing. I think they use DSS to induce. Oh, sorry, I tried to. I find it's harder to. Use that. All right, I think, can you hear me? Yeah? All right. So what happened, and they did immunohistology study, found P2X7 and Pannexin-1 all present in meningeal ganglia of the mouse. And when they use DSS to treat the animal, they find the neuronal loss, okay, in these models. But then they gave OATP, which is not very selective, but still can inhibit the P2X7 receptor. They can prevent the neuron loss, right? So then they actually count. They count how much neuron lost, and you can see here if they gave P2X7 inhibitor, they can prevent neuron loss, and also they did some study here for the prebenazine. It's Pannexin-1 inhibitor. Also, the neuronal loss is not as severe as the just DNS treated. So, which is very interesting, encouraging study, and so that's why I think we probably better to have a look what happened, and not just in the animal model, what happened to the human tissue. So that's the I did research. My PhD student did a research. So she actually tried to have a look at the profile of Pannexin-1 and P2X7 receptor in human colon, and also want to have a look if Pannexin-1 really is the ATP releasing channel in these tissues. And also we want to have a look if Pannexin-1 and P2X7 is involved in the pathophysiology of IBD. All right, so for as I mentioned, the specimen I have a really long-term collaboration with the colorectal surgeons in Sydney, and we have a specimen from colon cancer. So we usually, because colon cancer, the doctor usually section quite large colon, try to away from the colon tumor sites. So we actually took about 10 to 20 centimeters away from the tumor sites, so we use as a control. And we also get specimen for Crohn's patients and also ulcerative colitis. And for the tissue processing, we usually want to have keep intact the tissue doing immunohistochemistry, and for doing probably I just have this one out here. So for real-time PCR and Western, then we want to usually separate it, only just take about the mucosa part and also the muscle part. And then if we're doing the mucosa explants and to develop inflammation models, we actually just take the mucosa part, but we want to leave as much as submucosa as possible, because that's to have look if any leukocytes, lymphocytes infiltration can occur. So that's how we separate our tissues for the mucosa part and the submucosa. Submucosa contain lots of blood vessels, and also submucosal ganglia, which is a group of neuron cell bodies. And here is circular muscle, and then there's the mesenteric ganglia, and then that's longitudinal muscle, and this part is serosa. All right, first we actually did some immunohistochemistry study. So you can see it's harder to see, but the green color and on panel A, it's Panexin-1 immunostaining. You can see Panexin-1 actually stain around the epithelial cells, and so that's P2X7 as well. Okay, so on the this luminal epithelial cells and the crypt epithelial cells. So that's for its colon tissues, and also we had looked at KCO2 cells. So this is a cell line developed from the epithelial cells of colon cancer patients. And you can see Panexin-1, it's the green color, it's immune around the KCO cells, which means these cells are kind of on the cell membrane, and of this channel on the cell membrane is a function channel, and the Panexin-1, and for the same KCO cell also express the P2X7 receptor. So then we actually, to measure ATP release, and use the biochemistry method and to see how much ATP release. And also, we know the ATP release, one of the major function is to sensitize the P2X3 receptor on the sensory nerve ending to trigger peristalsis. So that's why we think that, of course, obviously in the colon or intestinal system, distension mechanical stimulation is one of the mechanisms to increase ATP release. So what we did for the mucosa strips, we put in organ baths and give a certain tension, and to increase the tension, so mimic the stretch of the physiological condition. So you can see, and when that's the basal ATP amount when we doing stretch, and with the time, there's a time course. After 2 minutes, 5 minutes, and 10 minutes, we collect the bath solution and measure how much ATP release. You can see as a substantial increase ATP release in response to stretch. And carboxylone, which is not very selective, it's a blocker both connexin 43, 45, and also Panexin-1 as well. But to start with, we usually use blood inhibitors. So you can see the carboxylone can reduce ATP release. So that's in the colon mucosa. And for the cell line work, we use a hypotonic solution, which is 50% less sodium chloride compared to the normal Krebs solution. You can see that's hypotonic also can increase ATP release, and this action also can block by carboxylone. So we think it's Panexin or connexin channels are the ATP release channel in the human colon. So what about Panexin-1 expression in IBD? And we did a real-time PCR study, and you can see Pannexin-1 messenger RNA expression, it's dramatically reduced in ulcerative colitis and Crohn's disease in smooth muscle, but not much change in mucosa, which is weird. We just found out why is that? Especially for ulcerative colitis, we always learned from literature it's just constrained in the mucosa, not transmural, right? So for Crohn's disease, of course, it can affect all the layers from mucosa, submucosa, and muscle. So that's no surprise if any change in Crohn's disease. But it looks like ulcerative colitis change a lot. So that's the messenger RNA expression. So then we also have look at the Western to have look at the protein level. So you can see smooth muscle and mucosa ulcerative colitis, no change at all. And but Crohn's disease still we can show the reduction in protein level. So apparently messenger RNA and the protein is kind of mismatched, not exactly tell us the same story. So then we had to look at reaction. You can see the brownish color is Pannexin-1 staining. It's really profound staining. All the cells looks to have a positive Pannexin-1. So which means really important, probably the ion channels for ATP release in the intestinal system. So that's the control. And you can see ulcerative colitis and really damaged mucosa, and also for the Crohn's disease, also very damaged, and they are very positive staining for Pannexin-1, and also these leukocytes have positive staining as well. And you can see for the ulcerative colitis here, its angiogenesis looks like occurred. Lots of blood vessels in the lamina propria and some mucosa as well. So have a look at the blood vessel here. And so this submucosa, lots of blood vessel, this is normal control, and the blood vessel really is round and normal. But for the ulcerative colitis, you can say irregular. That's the symptom of a swollen tissue as well. You can see the red blood cells. It's very positive staining for Pannexin-1. We really think the mismatch by messenger RNA and the protein, maybe it's caused by the red blood cells. Because red blood cells, we cannot measure messenger RNA, but can measure protein. So that's really, it's kind of give us a bit mismatch result. All right, so then we also find interesting ulcerative colitis. Even you said we all know it's not transmural disease, but in myocardial ganglia, ulcerative colitis have very weak staining for Pannexin-1. So which really prove some of our previous study, we find ulcerative colitis have motility change a lot. It's not something to do with the damage of myocardial plexus. And you can see here the Crohn's disease and lots of blood vessels, positive staining Pannexin-1 in serosa area. And this serosa is really in the thickness much increased compared to normal tissue. Right? Normal tissue is really quite thin. All right. So what about P2X7 receptor expression? So we find the messenger RNA, we can say not much change at all. Only slightly see the smooth muscle have a marginal increase for the P2X7 in ulcerative colitis, but not much. And also for the protein, Western, we didn't see any change in the IBD. All right, so then we have looked the immunohistochemistry for P2X7. We can see all the cells almost have a positive P2X7, and we can't see any difference between the disease compared to control. So no difference at all. That's the impression. And so then we actually adapted to the model for the explant colitis. So we actually use TNF-alpha and interleukin beta to incubate with the mucosa from the control patients. and for 16 hours. Because when we follow Scott-Smith method, we culture to 20 hours. But 20 hours, after 20 hours, the non-TNF-alpha treated control, also the epithelial cells starting falling. That's why we did time course. We found the 16 hours is the best. So it's 37 degrees and 95 supply have to have a sufficient oxygen. And we also use Pannexin 1 inhibitor, 10 Pannexin 1, and also P2X7 antagonist, A438079, to see if these two inhibitors can reverse the TNF-alpha interleukin-1 beta induced colitis. So that's what we did. So you can see here on the top panels the incubation control. It's looks like the crypts and the luminal epithelial cells quite intact, not much damage. But when you treat TNF-alpha and interleukin beta, really very damaged. Almost all the epithelial cells falling off. So then we actually treat 10 Pannexin 1 and slightly kind of to prevent TNF-induced damage. And P2X7 antagonists have a very good result, prevent the inflammation. And if we put the 10 Pannexin 1 and a P2X7 antagonist, really didn't improve much. Okay, looks like the best result is P2X7 antagonist. So then we did this is really only n equals 4. We can't give much conclusion yet. So we counted for the percentage crept damage, and you can see the TNF-alpha and interleukin beta treatment have a really big damage, very significant, and P2X7 can inhibit the damage almost back to the incubation controls. We also did the luminal damage. Luminal damage looks like quite inconclusive. So next thing we want to do is to test the lymphocytes infiltration, and also we want to measure COX. Okay, COX-2. We know the prostaglandin COX-2 is very important. We want to measure that, and we know the TNF-alpha interleukin-1 beta. That can also trigger some chemokine release. We want to measure the chemokine as well. All right. So now we did this K-code to measure trans-epithelial electrical resistance. So we know the epithelial cell can form tight junction, so can produce resistance. So if damaged, the tight junction damaged, resistance will reduce. So you see here, this is the control, and here the red color is TNF-alpha interleukin-1 beta. You can see resistance really dramatic damage reduced, which means the epithelial cell tight junctions damaged. So when we give carboxylone and P2X7 antagonists, you can see this damage can be reversed. So which is a really good sign as well. Okay, summaries. So TNF-alpha and interleukin-1 beta is the major cytokines are known to be elevated in IBD with a capacity to induce the mucosa chemical expression in human colon. Okay, so treat healthy human mucosa explant with TNF-alpha and interleukin beta can generate a form of mucosa damage and inflammatory response resembling to the colitis. And the colitis explant may be a useful model for the development of IBD therapeutics. Okay, so also we found the Pannexin 1 and the P2X7 receptor may participate in the development of inflammation. So this is a kind of the diagram. I'll explain what happened. So Pannexin 1 and can mediate ATP release, and then can activate enabling P2X7 receptor. Then Pannexin 1 and P2X7 may form death channels, and they also can cause the tissue damage. Damaged tissue can release large amount of ATP, and that can stimulate cytokine and chemical release. And that's plus microorganism in the lumen can further damage the epithelial cells, make the tissue inflammation occur. So that's our theory. And if we block pan-X7, especially block P2X7, we probably can inhibit the tissue damage. So to conclude, and the pan-X1 in the colon may function as ATP release channel to regulate mechanosensory processing and intestinal blood supply, gut hormone secretion, and neuronal transmission and motility. And the pan-X1 channel and P2X7 receptor may play an important role in the initiation and the development of intestinal inflammation. And inhibition of pan-X1 and especially of P2X7 probably can represent a novel therapeutic option in the treatment of IBD. So that's my group, and so I have the majority work conducted by my PhD student Erica Diazemos, and so that's I also have collaborations with colorectal surgeon from St. George Hospital in Sydney. Thank you very much.
Thank you very much for this very interesting talk. Is there any question from the floor? No?
Oh, thank you. Very interesting talk, and thank you. And according to your results, the release of ATP should activate this receptor to promote more inflammation, right? And it may be mediated through the pan-X1 channel. How do you, during acute inflammation, when there's a massive damage of the cell, how do you discriminate that this is mainly due for the release of necrosis or cell death, that maybe the channel can be a pathway leading a physiological release of the ATP, but not maybe in acute inflammation of the mucosa? That they form like a complex, these two entities, molecular entities, for like a complex, that they are close, or they are like separate?
I think there is a study, but it's not, sorry. Yeah, there is study for indicated they form they kind of very close associated together. If only put one, like I would say, the cells I showed, if they only put P2X7 or only put pan-X1 individually and deploy a large amount of ATP, it's nothing, it's almost like a normal. But if they put these two together, and I think they had a look, and it looks like they very close associated, these two, and the cells started leaking. That's why the theory is. But that's only for the kind of cell lines and oocyte research. If this really occurred in the tissue in real life, that's the further study we want to conduct. Yeah.
Thank you.
Yeah, thank you.
You also mentioned the release of IL-1 beta and caspase 1
Yeah.
that is influenced by your receptors.
That's right. I think it looks like for Pannexin-1 it's a non-selective channel things. It's not just release ATP, they also release actually interleukin-1 beta and during inflammation.
And I'm sorry to interrupt you, but do you think it's influencing inflammasome activation as such?
Yeah.
Or do you see whatever IL-8 secretion from these cells as well?
I think it's probably during inflammation. Yeah,
that's really interesting.
Yeah, that's right. And also, and the next things I actually think my research also interested in non-neuronal acetylcholine, and I actually think maybe acetylcholine also released from the Pannexin channels. And somebody reported said prostaglandin E2 and it can release from the prostaglandin from the Pannexin as well. Yeah, but I don't know because it's anything below than 1 kilodalton probably released from Pannexin. Yeah.
Okay.
Yeah. Yeah.
Thank you for your excellent talk.
Thank you.
I didn't notice very well. Your research was, you said, on surgical specimens, yes, from patients with inflammatory bowel disease. How did you select this? There were patients treated with something before. There were patients with have only conventional and some anti-TNF therapy. How do you select the specimens for your research?
That's a very good question because when we publish data, often the reviewer asks this kind of questions of how you. So every time we actually collect the specimen, then conduct the experiment. So then we need to go back to doctor, have a look at all the patients' history and which drug they use and what stage. One episode just very acute, or they relapse from the chronic, or the remission, how long. So we have to put all this information in kind of a table to when we publish the data. And at this stage, we really can't control. And some actually, you can tell it's the patients and very acute, looks like a very bleeding. Some is kind of chronic inflammation, but just relapse. And the treatment can be different as well. Most of patients have, like Crohn's disease, have infliximab and also.
Yes, but do you think that previous treatment can influence the results of your research? Because some of them could be treated with anti-TNF, some of them with anti-integrins or something like this.
Yeah, that's possible.
Or some of them, did you do research only also on the naive patients that they didn't have treatment before?
I think we really can't do much. We don't have much choice because nobody wants to contribute. Yeah, that's right.
Going without any treatment before.
Yeah, we do have some. We do have some of the patients. They have just acute and all of a sudden really emerging situation. The doctor has to have operation. They have no treatment. We do have these specimens as well.
Okay, thank you. And the second question, what do you think, in your opinion, that will be the most interesting direction in molecular research in the future years for these patients with IBD?
Okay. So I actually see, because I think a P2X7 receptor can be interesting because it's a clinical trial use P2X7 receptor antagonist to treat other inflammation disease like I think it's some rheumatoid arthritis, and they're on clinical trials. And but for the advantage for treat colon inflammation, I know it's lots of side effects can occur with these compounds, but for the advantage for colon inflammation, if we have developed some compounds only have a local action, not absorbed into the body, so that can avoid side effects. That's how I feel. It's probably can for pharmaceutical industry can be a direction to go. Yeah.
Thank you.
Thank you.